Okay, so as a first real part of the course, I'm going to present you really an overview of Bgee, what's in it, what it does, okay? So as we already said, Bgee is the work of the team, so us two are here presenting it, but really it's a team effort. And if you contact us on any of these in any of these ways, the primary way is the email. Different members of the team will answer depending on the question and the expertise. So what is Bgee? Bgee is a database of gene expression, and our goal is to understand gene expression. So gene expression is a very complex thing. Genes are expressed in different ways, in different places, at different levels, and we want to make this useful. So we want to understand gene expression ourselves in an expert way, and then make it useful to biologists. So that's really the goal of Bgee. And now I'm going to do a rapid demo of Bgee up just to show you a bit around the website. So this is the new website we have since 2 weeks. You can see here different tools on top, and here a big gene search button. So then you get to the primary information you get in Bgee. For example, if I search for insulin, I get all the genes which are insulin, and we sort smartly so that the first genes are the insulin genes and not, say, insulin receptors, and you have the genes in the different species. So for example, if I click on insulin in human, you're going to see the page of insulin in human. And here, what's important, we integrate all types of data together, so to give you one summarized view of expression. So here you see that you have microarrays with an A, bulk RNA-Seq with an R, and so on. And here you have, so SC will be single cell, don't appear here, in situ and ESTs. And here insulin appears on top in type B pancreatic cell and body of pancreas, which is indeed where insulin is primarily expressed. And you see a big jump in our expression score here, down from almost 100, 100, almost 100. So we integrate all the types of data together, and then you can see the biological information come up. And we also have the comparison between species. So here I have orthologs of insulin in different species. And if, for example, I compare among mammals, so here the line mammals, I will see the expression comparison among mammals. This would take it's not the fastest thing, so I'll just show you another page with Bgee just to show you something else. If I go to again insulin, for example, and I go to the mouse, for example, or the where I wanted to show a fly gene. Oh, I wanted to show you also our search allows that if you search even a short term, it finds the right gene. And if I show in fly here, you see that here we've integrated the fly cell atlas. So you'll have also integrated in the same way as you would have microarrays or bulk RNA-Seq, single cell RNA-Seq. So here you have the expression of photoreceptor in the insect head. So very specific expression. And now I'm going to open the next WOOC lab. Sorry, I didn't finish showing you things on Bgee. So this is the comparison between species, and here you see between mammals, I have the islet of Langerhans, the underground pancreas, the pancreas, which are top as conserved expression between mammals. So you have this expression which is not just in one species, but between species and conserved. And finally, what species do we have in Bgee? We have only animals. We aim to have a great diversity of animals. Right now, we don't have all the animals. I mean, we have those which have species. So if I click here, I have the photo of all the species we have. So you see a lot of domestic, model species, of course, a lot of domestic species. We had a focus on this, quite a few fishes also, a few insects, and we curate the data so that we only have wild type healthy, so no cancers, no mutants, no knockouts, so that you can know that the gene expression you see here is the normal gene expression comparable between species. So what do we do? We take the data from a diversity of sources, so microarray data, which used to be the main source of gene expression data large scale, bulk RNA-Seq, which has become the main source of gene expression large scale, single cell RNA-Seq data, which is increasingly an important source of data and has much more fine detail than bulk RNA-Seq, but of course has different challenges that Frédéric will present later. and also in situ hybridization data, which is less large scale, but has very fine levels of detail and can be very important for model organisms. And we take this from the different database it is, so we don't restrict ourselves to one source of information. And all this data we curate to make sure that it is healthy, wild type, and it will be annotated in the right way. We process it so it is all comparable. We can bring together all this gene expression data to give you something like the gene page I showed you. We map it to ontologies. We will explain a bit later what are ontologies, don't worry, but to make it all integrated and comparable, and everything is integrated together. So it's really important to take from this page is that we take different types of data from different sources and carefully process them to integrate them all together in one database, one view. So it's not like other databases will have separately a bulk RNA-Seq and a single cell view, separately the in situ, separately the RNA-Seq. In Bgee, everything is together. I've spoken several times about curation, so I'm going to speak now a bit about what is curation and why it is important. So there are two types of database in the life sciences, uncurated and curated. Uncurated database, the typical example we all know is GenBank or NCBI nucleotides. There is huge redundancy. You look for insulin and you'll find 50, 100 copies of the insulin sequence. There's low organization of knowledge. People might have put details about the gene, the information, the genome or not. You don't know. But the main added value, it is up to date. So you submit sequences to GenBank. They are online the next day, and everything which was published is there. So it's complete, up to date, but poorly organized, highly redundant. Conversely, curated databases, the data is verified, the redundancy is minimal, is managed so that you have one time the insulin gene. Annotations are standardized so that you can easily query things and compare them. And the main added value is that the knowledge is organized and reliable. And the typical example everyone knows is SwissProt, the curated part of UniProt. So when you go to UniProt and there's a gold star that's curated by professional biocurators at SwissProt, and everything is standard and comparable. And I just put on the bottom of the slide the definition of biocuration from the International Society for Biocuration. And I think what's important here is that it's translation integration of information relevant to biology that enables integration of scientific literature as well as large data sets. So really we want to translate the knowledge and make it useful. That's really what biocuration does. And in biocuration, one of the things we do is annotation. Annotation is when you associate a biological object to a feature based on evidence. That's a bit dry. So for example, you have a gene ontology term, say you say hydrolase, and you have a gene and you associate the hydrolase term to the gene. So now you know this gene is a hydrolase. And you can do this based on the blast hit. So this gene, its first blast hit was a hydrolase. So I say, hey, it's a hydrolase. That would be an annotation, but not curated. You didn't check anything. Or you read a paper where someone did experiments on this, on the product of this gene, saw it had hydrolase activity. And from reading this paper, you put this annotation in the database, you associated this gene ontology term with this gene. In this case, it's a curated annotation. And Bgee, why am I telling you all this, is a curated database. So all the expression data we integrate is verified. Data which doesn't fulfill our criteria of being healthy, wild type, and following protocols that we can use is excluded. And every expression data set is annotated by manual curation. So we have biocurators who read all the metadata of the submitted data. They read the paper, they read the supplementary data of the paper, and when information is missing or contradictory, then they contact the authors. And all these annotations follow standards, and those standards themselves are curated by biocurators. So for example, the gene ontology, you all know, is made by biocurators, and all those standards we use are curated so that we have the highest quality and the best integration of data. So we, as I've told you several times, only take wild type healthy. So why do we take only wild type healthy? That means no experimental mutants, no knockouts, knock-ins, CRISPR modifications, and no cancers, diseases, and so on. Why do we take this? Because it is informative on the causal function of genes. What does it mean, the causal function of a gene? Well, causal function is, for example, the heart. Its causal function is to pump blood. It also has other functions like having heart attacks or making noise, but this is not the function which is important for the heart. Why it is there is to pump blood. In the same way, you want to know that a gene has a function which is the function for which it was selected. The function of a gene is not being dysregulated during cancer. So that's what we want to get. It is also evolutionarily relevant. So if I want to compare, I showed you the comparison of insulin between mammals, and you saw that it was expressed in different mammals, always in type B cells and pancreas. If I compare a human tumor to a knockout mutant mouse, there is no information on what does this gene generally do on mammals, what they do in the ancestor. And it also provides a reference for biomedical studies. So we saw many of you come from a biomedical interest and background. So in biomedical studies, you will often be interested in disease states, but you need to know what can you compare this to? What would this gene do when there is not a disease? And Bgee will provide you this reference. So an example of looking at wild type healthy, so some of you actually cited GTEC. So GTEC is partially curated. It is a very large data set of bulk RNA-Seq from humans. And in the description, they say that they collected non-disease tissue sites across 1,000 individuals, but they did not exclude specific donors from specific tissues based on their cause of death or medical history. So the biocurators at Bgee, we got access to all the detailed pathology reports and annotations of each sample and re-annotated them. And we found that there were many issues that these samples were not healthy. So if you take the subjects, you have a small half of the subjects where we accepted all the data, they were healthy, but you also had a third more or less of the subjects which we rejected because they were not healthy. For example, people who died of cancer, people who died of drug abuse, people who were BMI above 35, which is defined by the World Health Organization as morbidly obese, and so on. And in the samples of those which we did not completely reject, you also had, for example, people who had dementia. We did not reject the other tissues, but we did reject brain tissues as not being healthy. You had people who had liver disease or ascites, which is another liver disease. And in that case, we did not keep the liver data. So what you see is that when you take GTEC data from GTEC, you're getting gene expression, which is a mix of how the genes are expressed in the healthy tissue and how it's expressed in either the diseased tissue or in people. For example, if you died of a drug overdose, your physiology is very messed up. So the gene expression is all over the place. And if you take the GTEC data from Bgee, you will have only half the data, but you will have the purely healthy data. So you'll know this is how the genes are expressed in healthy humans. So in Bgee, we reviewed all the GTEC version 6. There's now version 8, which we have not yet re-curated and released. Out of 12,000 libraries that were reviewed, it was a lot of work. We kept 5,000, so less than half. And we have access, because we have the private access to the restricted data, to the exact age, ethnicity, and so on. But GTEC is sensitive data because humans, so we only release broader information. We're not allowed to give all the information. You can go to your annotation pages and to Bgee experiment pages, which Frédéric will show you later, and see all the data from this specific big experiment. Another example where we recurated data is the fly cell atlas. So this also was in principle a curated data set, but we recurated it in collaboration with specialists of fly single cell and found quite a few errors in the fly cell atlas, which we corrected. So if you access the fly cell atlas in Bgee, it is corrected. For example, you have the male reproductive system, which was reported with different identifiers. So if you query one identifier, you don't find all the data from the male reproductive system. There were inconsistencies, so they would report an ovary cell in a male or a testis cell in a female. And there were places where they should be reporting a cell type, where they reported an organ, which is not consistent and doesn't allow you to report the right thing. So we recurated and cleaned this up. And for this, we curated 1,500 conditions. And in the end, we have 27 million processed expression values that again you can get from the experiment page of Bgee. Then the curation, apart from selecting the data, allows us to standardize the data. And data, when it is standardized, it is reusable by everyone. It means that you can query the right terms and find the data you need, only the data you need, and all the data you need. And standards allow us to define what is the metadata. Metadata is like if the data for a gene expression experiment is how highly the genes are expressed, the metadata is in which organ I took this, from which strain of mice, under which conditions, and so on. We, as I told you, read the papers as well as the metadata from the databases, and when information is missing, the biocurators contact the authors. And then in Bgee, you can find this information standardized. So here you have a description of the experiment and the link to the article and to the original source, and this we have recurated. And then you can get the standardized data processed in a standard way so that you can integrate this, for example, in your analysis in R. And also for those who use single cell, we, for all single cell experiments, provide the H5AD files, which are standard, which is used in single cell analysis. And we, because this is not something everyone knows, we provide a documentation on our website about the H5AD, what is in it and how you can use it. For example, here is how to use H5AD in Python. There is also standard R packages to use H5AD, which will provide the documentation.
I got a question.
Yes.
On the single cell data sets that you provide on the Bgee database, did you also review and reannotate cells that might be mislabeled and maybe filtered out cells that were difficult to label, for example?
So we do not, we trust the author's decisions on what are the cell populations. Because this is something where we curate, but we have to trust the experts who published the data.
Okay. Thank you.
Single cell RNA-Seq is more complicated than bulk RNA-Seq. Any of those among you who manipulated it must know this. And one of the things is that we have a feeling sometimes that every second paper about single cell RNA-Seq introduces a new protocol. So we have classified all the protocols which were found used in the literature on how they work and what consequences they have for analysis, because some protocols will give you access to the full RNA and some only to a part of the sequence. Some will allow you to see which sequences are present but not really absent. Some will allow you to see all types of RNA and some only protein coding, and so on and so forth. And so we have classified all this. Right now we have 4 protocols that we accept in Bgee. Every new protocol we have to do benchmarked pipeline, so it's a bit we try to do this very carefully, and we focus on the ones that are the most used. And so we have this page you can find on our website with all those protocols classified. So this is a service to the community from our biocurators. Again, we do not have all of these in Bgee. We only have 4 in Bgee, but if you can look at this and find information about all the different protocols of single cell RNA-Seq. And then when we have this data that we have curated and find the metadata, we annotate it in a standardized way. And we annotate to ontologies. What is an ontology? An ontology is first a list of terms, so we agree on what words we use. If you just have a list of terms, you have a controlled vocabulary. Then each term has a definition. If you have a list of terms with definitions, you have a dictionary. And what makes an ontology is that you have not just terms which we agreed on and definitions, but relations between them. So for example, if you say the cerebellum is part of the brain, this is a relation between the term cerebellum and brain. And then when you have this, you have an ontology. You can have different types of relations. The cerebellum is part of the brain. The cerebellum contributes to the nervous system and so on. And they allow automated reasoning, which means I can do complex queries which would know if I want all genes expressed in the brain, I should take the genes which are expressed in the cerebellum. So the example everyone knows of ontology is the gene ontology. Here you have, so you see here, for example, cell migration in hindbrain, and you see that it has relations with other terms. So it, and there's different types of relations, is a part of regulates. So cell migration in hindbrain is a hindbrain development and is a cell migration. And then this can be used to standardize information. For example, here you have a Hox gene in zebrafish, which has this function annotated because it is involved in this biological process. And so this way you can recover all the genes which are involved in cell migration in hindbrain. But if you wanted all the genes which are involved in nervous system development, you could look for that and you would recover this because you could reason that cell migration in hindbrain is part of nervous system development. We of course use the gene ontology for annotations, but our core focuses on anatomy because that's where gene expression is important. Where is a gene expressed? And for this, we use and co-develop with other teams the Uberon, the Uber anatomy ontology, which describes the anatomy of different animals, actually of any bilaterian animal. And so here the terms, rather than being function of gene, would be anatomical structures. For example, here the liver. The relationships would be also is a part of, but also is it in some only some taxa? For example, you will find lungs only in tetrapod animals and so on. And so you have the liver, which is part of the endocrine system, part of the digestive system, only in vertebrates, part of the abdomen, and so on. And we also use the cell ontology, which is an ontology of all the cell types. So here, for example, you have a Kupffer cell, which is a hepatic sinusoid, which is part of hepatic sinusoid in the Uberon and is a tissue resident macrophage and has these different relations, also relation to a Go term because of its function. And single cell is again complicated because the cell ontology does not manage really to keep up with the huge diversity of cell types we are discovering from single cell. So for example, you have T neurons of different types, which are all annotated to the same cell ontology term because we don't have the level of detail. So, and we always knew cell types. For example, last year they discovered the difference between octapaminergic neurons and teraminergic neurons, but they were not yet distinguished in the cell ontology. So this is a challenge that everyone in the single cell annotation world has to live with. But we use it as much as possible, and we complement it with capturing the free text annotation of the authors so that if they had more detail in their free text, we can capture it. So what makes an ontology useful? It's a common standard which is used across many different ontologies. Resources. So you know that the gene ontology will find it in many different databases and resources. Uberon is used, it's common to all animals and is used in all the large projects which sample different organs and cell types in different species, whereas the GTEC or human atlas in human, the flies atlas, and so on. It covers a large domain of knowledge. The gene ontology is all the functions of genes. Uberon is all anatomy of animals. And there are many tools leveraging it. So you all know gene ontology enrichment tests. We will show you some tools today which leverage anatomical ontology. So what do we annotate? We annotate gene expression to anatomy. This is what I've been speaking about. So anatomy is both the anatomical entity and the cell type, as I showed you in the examples earlier when I demoed. We also annotate to developmental and life stage. So you want to know that this gene is expressed in, not just you want to know in the liver, but you want to know, is it in the adult liver? Is it in the juvenile liver? Is it in developing liver in the embryo? Is it in a old liver? If you're studying aging, you will find that in Bgee we annotate specifically the aging process so that you can find old mice, old humans, old worms, and so on. Sex, because many genes have different expression depending on sex. So male, female, in some species it's a bit more complicated. You have hermaphrodites and so on. And when we have the information, strains for, so it's called strains for lab species, varieties, or races for farm animals, and populations for humans. When we have this information and are allowed to share it, we also have it. So this is an example of a notation. This is one big single cell data set, and this is how it appears on our data on our web page where you can download the data. So you have the library's ideas, of course, and then you have, for example, the authors call these cells these different terms, which are not always super clear. So you see BP, endo, HC, and we reannotated this to the standard terms of the cell ontology. So BP is retinal bipolar neuron, endo is endothelial cell, HC is retinal horizontal cell, and the same for the anatomy. They wrote these different terms, and we reannotate them to these standardized terms. So you can see all this is in the macula lutea. And we also annotate the age, as I told you. So here you see the age, so that if you were interested, for example, to get only single cell from humans above 65, you would recover this because it is part again of an ontology. So you could do reasoning on it and recover it. And here the sex. So in summary, we curate data from these different sources to identify valid data sets, so technologies we can use, healthy wild type samples. We recover all the necessary files and this is pretty simple for microarrays and bulk RNA-Seq. It's just the expression data, and for single cell we also recover the BARs, the BAM files, and the association between barcodes and cell populations. And when this is missing, which is alas too often, we look at the supplementary data, at what they put in Zenodo or Figshare, and we contact the authors. And if they don't provide it in the end, we cannot integrate it, but that means you couldn't use it either. It's not usable. Then we annotate this based on the protocol used and the paper, annotate to the ontologies of anatomy, cell, development and life stage, sex and strain. We quantify the expression, determine what is the state of expression, is it expressed or not. Frédéric will present you the details of this, and then we integrate all this into Bgee. So that is the end of part 1.